let's go to our second of 3 chapters for the muscle. This one's going to really talk about how a muscle contracts, and there's really 4 different steps, going from excitation of that muscle to eventually relaxation. And we're going to cover these in detail. There are some things that we're going to cover a little more in detail when we do our neuron chapter. So there's going to be some things I'm going to kind of go glance over this time, but later we'll go into it for our next unit. Basically, these are the 4 steps of muscle contractions. There's excitation. The muscle gets a signal to do something. Excitation with the contracting couples. Contraction. And then relaxation. And what I'm going to do for the next slides is every time we do one of these 4 things, I'm going to have it with a bullet point. 1, 2, 3, or 4. This first video is only going to be on excitation. So when excitation occurs, this is occurring because the neuron is going to stimulate the muscle to do some sort of movement. And that's going to be an action potential that moves along the somatic motor neuron to the muscle fiber. So let's say there's a muscle right over here. The neuron, which is going to be in the central nervous system, is going to have this axon. We're going to talk way more about these different parts in our next exam. So let's not get too bogged down in this detail. But basically what happens is a current is going to move down the neuron to these little ends called terminals. And that's going to be in contact with a muscle. Where those little contact ends come from that neuron, is going to be called a Neuromuscular Junction. So this is where the neuron and the muscle comes together. So this will be the muscle, this is going to be the neuron, and where there's going to be a couple of different steps that occur, that's going to send electrical energy into chemical energy back into electrical energy into the muscle. And that's what we're going to cover. I'm going to hide a couple of things from you because we're going to cover them even more in detail in the next couple of exams when we talk about the brain and neurons itself. But we'll come back to those. Keep going with the anatomy of this area. There's the axon terminal. So here was the axon. This is going to be where it ends, and it sort of comes together with our motor end plate. This wiggly line right over here is called the motor end plate. Think about motor. Motor is usually going to be on a muscle, and it's the end plate where the neuron is going to send a message to the muscle, which will eventually get relayed to do some sort of action. The synaptic cleft is this opening in between the motor end plate and the axon terminal. The synaptic vesicle is going to be these little bubbles that are in the synapse. When an electrical current comes in, these calcium channels will open up and allow for these vesicles to do this thing called exocytosis. Exo means to exit. Cytosis means sort of this little bubble that it's formed. And what's going to happen is these bubbles will sort of merge to the end of the axon and spill out these neurotransmitters. And these neurotransmitters are going to go into the muscle and then do some sort of action. Again, I am hiding a few little things from you because we're going to cover them again when we do the nervous system. What then happens is these voltage gate calcium channels open. So I think I already sort of mentioned this, but an electrical current comes through. This electrical current is going to open up these little gates. What's going to sneak in is calcium. Calcium is going to start an action potential. Action just potential means something is going to occur, and it's going to be usually muscle stimuli. And then calcium is going to bind to the synaptic vesicles and trigger the release of these neurotransmitters. So let's kind of get this all together. So we have our neuron. Electrical current comes through. It's going to go ahead and open up these calcium channels. When calcium starts to flood inside of the neuron, these little vesicles that hold neurotransmitters are going to basically come to the end of the axon and spill out these neurotransmitters. And they're going to actually go ahead and start to get binding and into the motor plate. Next is going to be these ligand gate sodium channels over here. They're going to open when the neurotransmitter is called acetylcholine, and that neurotransmitter is going to go ahead and enter the motor plate area into these ligand. Ligand just means it's an area that's looking for one item, and they're ligand gates that let sodium in. So let's go over a little bit what we've learned. So, quick anatomy, here's that motor plate. The synaptic cleft is basically the distance between the axon and that motor plate. Here is the nerve. This is called a neuromuscular junction, because this is the muscle, this is the neuron. What's going to happen is electrical energy is going to come down in the form of this action potential. We're going to talk about how this gets generated in the future, but let's just say for now, this is a message that's coming from the spinal cord or the central nervous system and is traveling down this particular neuron. What then happens is this triggers our first thing to occur. It's going to open up these calcium channels to allow calcium to come in. Once calcium comes into the axon, we're going to go ahead and have calcium bind to these vesicles, and these vesicles are going to go ahead and merge to the end of the axon and release. ACH is just shorthand for acetylcholine. Acetylcholine is a neurotransmitter that's going to be able to go across the synaptic cleft and bind to these receptor molecules. What then will happen is they're going to go ahead and, well, let's do it in our next picture. So when acetylcholine gets released into the synaptic cleft, what happens is it binds. It binds to these ligand-gated sodium channels. Now, don't let these names sort of trick you up. I know when I was a bio student, these names really freaked me out. But let's just kind of break it down. Ligand just means a area that only is looking for one thing. And it's a gate. So that means this gate could be opened or closed. And what's the key for this ligand gate? It's going to be sodium. So it's going to be acetylcholine. And when acetylcholine opens the gate, what's going to rush in is going to be sodium. So here we go. We have acetylcholine binds to the ligand gated NaCl ligand. Again, it's just a gate that's opened up by one thing, and that's going to be the acetylcholine. When the gate is opened, what's going to rush in is sodium. Sodium is going to rush in from the synaptic cleft into the muscle. And then from there, it's going to do this thing called depolarization. This is going to be happening at that end plate. Remember, the end plate is this area where the muscle is. And it's going to allow us to do our next event that we're going to cover. But before we do that, let's answer this question. If enough ligand binds, sodium channel opens up and depolarization occurs. So a threshold was created, and what happens when the threshold is reached? When that threshold is reached, it's going to equal an action potential. And the action potential is what we're going to cover in our next thing, excitation.